Okay, good afternoon, everyone. At least good afternoon to those who are in Europe. Maybe we also have some participants coming from the Americas, and for them, then is the morning. My name is Andrea Rosati. I'm the Secretary General of the EAAP, the European Federation for Animal Science, which is the continental organization of animal scientists, the network of, dealing with the livestock industry and animal science. You are very welcome today to come to this number 27 webinar organized by EAP. The title is quite clear and easy, Application on Genomics in Animal Breeding. We have three very outstanding speakers, and Alessandro Bagnato from University of Milan, Sebastián Demyda Peyrás from University of Córdoba, and Tosso Leb from University of Bern. So the three of them will talk about their views and their experience in application of genomics in animal breeding. This webinar, like sometimes other webinars, are organized in collaboration with an industry, which is our sponsor today, is Neogen. Neogen is a good friend of EAP. They sponsor us very often. They come with us. And not only sponsoring, but they participate actively in our scientific network. Anyway, they provide an extensive range of agrigenomic services and solutions for food and animal safety. Neogen works alongside breed association, genetic evaluation providers, companion animal breed registries, researchers, and individual farmers. So thanks very much to the Neogen people giving their trust to EAP and to the activities that we are doing. And thanks for making this webinar alive. This is the program that we have today. We'll have only 10 minutes to talk, to introduce. Maybe I will be shorter. And every speaker that you can see here, they will have 30 minutes each. I suggest them to finish before 30 minutes so that they can have some immediate question and answer. In any case, we have 20 minutes at the end of the conference, of this webinar. So from 4:40 to 5 o'clock. For the wrap-up session to final remarks and more questions. You can ask questions directly and also, through using the chat that is available here. I will spend a couple of minutes not talking about genomics in animal breeding because you will hear a lot, just to inform you about the next activities of EAP. I take this chance to update you. We have some workshops going on. One is about the Central Eastern European countries livestock farming. And the workshop will be in Krakow, Poland, beginning of April, and 19 to 11 of April. Then we have another one, which is quite new for our EAP, dealing with companion animals. And we have a workshop with companion animals. The first one we have is going to be held in mid of May, 14 to 16 of May in Milano and Italy, and we invited the best speaker, best scientists around the world on this field, and we really expect a very interesting workshop. So please come. This is gonna be very, very interesting. Also, if you don't deal with companion animals, to tell you what is going on, at least in these two main species, cats and dogs. Then we have the artificial intelligence. Everyone is talking about artificial intelligence, and EAP decided to organize a conference on artificial intelligence for animal science. And this will be held in Zurich, Switzerland, between 4 and 6 on June. Even for that, we really invited people from China, from USA, and the best European scientists. So be updated, to be updated about the application of artificial intelligence in animal science. This is the right place to be. And I close with the most important event we have, which is the annual meeting, the 76th annual meeting of EAP. This year will be held in Innsbruck, Austria, end of August. Last year we were in Florence, 2 years ago in Lyon. We go around Europe, and we just closed last week the abstract submission for this conference, and we are close to 1,500 abstracts. So I suppose we will have very interesting and many presentations that in Innsbruck. So some information for the audience before I pass the word to the first speaker. After each presentation, as I say, there might be time left for Q&A. You can raise your question in the chat box, of course, and then I can read it, or the chair will read it. And other thing I want to say, the webinar will be recorded and will be further available on the EAP YouTube channel. And please go to YouTube channel of EAAP. There is a lot of information, a lot of previous webinars or plenary sessions, many activities coming on that we did in EAAP. So stay tuned with us, with the EAAP website and through our newsletter or the social media for heavy news about the webinars and everything else that's going on in the Animal Science Network in Europe. Having said this, I will stop to sharing the screen and I will go to present the first speaker, Professor Alessandro Bagnato. And I have some notes here by every speaker, but I feel shame to read notes about Alex because Alex and I know each other since at least 35 years now. So that says more or less how old we are. We were little boys when we start to be young geneticists in Italy. Then our career share, and you got the right one. He is a full professor, University of Milano, in animal science at University of Milano, and DIVAS, I think that's your department. He's leading research on animal breeding and genetics with national and international partners. He had a PRIM project in 2022 on genomic breeding values in Aosta, and that's more or less, I think, what he's going to talk to us today, and has chaired the educational board for degree programs since October 2023. Alex is a well-known animal geneticist around the world, animal scientist first, but also specialized in animal genetics. Everyone know him in Italy, in Europe, and also for his activities. I know he's well known also in USA, where he stays sometimes and part of his career over there. So, Alex, I will, or should I call you Professor Bagnato?
No, Alex.
Alex sounds more familiar for me.
Yes.
Alex's presentation title is SNP Genotypes in Cattle and Goats for the Management of Herds and Flocks. So, Alex, please, you have 30 minutes. Huh?
I will try to stay
Please do it.
on time. I know that you will choose me to stop me. Thank you so much for inviting me also jointly with the Neogen that I know is co-organizing the meeting. And yes, I will talk to you about what is the activity we do as a research group. I share the presentation. Please let me know if you see full screen.
Yes.
Okay. Okay. So the the idea is to share with you the work that we are doing on cattle and goats using SNP genotypes. We are also involved in sequencing, analyzing sequences, but the SNP genotypes that we are using is a large database that we own, and is providing us a lot of information that we are using to disclose many things in cattle, mainly dairy and dual purpose cattle. Let me see if I am able. Okay, I want to start from this slide briefly, half a minute. Our work is to provide animal food for humanity, and this is a topic, very hot topic, because when I was born age 60, we were 2.5 billion. Now we are 8 billion. We are expected to be 10 billion in a few years. So the need to increase the food amount, this now the finite resources, and the two main goals that we are dealing with in this field is to do selection to have efficient animal. Efficient animals means that they are more able to use feed, to use resources, to transform them in animal food. And this means less use of land, less use of resources, and sustainability. Sustainability because we have finite resources. This is the field, the large field in which we work. Very briefly, our goal, as all of you knows perfectly, is to identify individuals better than the mean and use them as reproducer and generate a new progeny that is better than the previous generation. This is all these reproducing, choosing animals, is genetic improvement. And we have to deal, as we know, with phenotype, genealogical information are the two basic information we need, with pedigree, and then we work in this topic. Now, what is briefly how we came into the era of genomic? Phenotypic selection started, say, with Mendel, with the formation of first airbook. The big change was in advancing quantitative genetics with computer. I started working with computers, and that was allowing us to estimate the breeding value on large number of animals. So to solve a system, very complex system of equation. But the other revolution was the genomic era, when the human genome was sequenced, and then we sequenced the genome of cattle, swine, poultry, and we produced technologies that ended up in a possibility to genotype animals with these SMP chips at a very low cost. Actually, to genotype an animal in cattle, at least in cattle, has a range of cost between probably 18 to 22, 23 euro. Something that is affordable also for farmers. And of course, sequencing is another part of the genomic area. That's a slide that I like a lot because show how the physiology is changing according with the selection. And the one on the left is a very famous picture of broiler after 40 years of selection. And the one on the right is a brown cattle, and that was when was before the selection for milk. As you can see, the shape and the physiology of this animal changed a lot. So the challenge for us is to apply selection using genomics to produce food in all the globe using less resources. And the agri-livestock genomics is playing a crucial role that is increasing day by day because it is becoming central. So in the genomic era with SNPs, we can do a lot of application of this information. Knowing what has been inherited from a sire and a dam, analyzing the DNA and the genome of a newborn allow us to follow along the generation the recombination events that occur during meiosis. And we can use this information to disclose variability across populations, to map structural variation on the genome, to identify run of homozygosity, to spot other information. And all this technology has been used firstly to predict the total genetic value using wide dense marker maps, as the paper of Theo Mewissen in 2001. So he, together with Ben Hayes and Mike Goddard, they published this paper 8 years before we had the sequence of the cattle genome and the possibility to apply this approach. But that was the first approach and the first application that we had in cattle, swine, and all the livestock species. And something that I found very interesting is the retrospective study by Guinan et al. In 2003. They were comparing on real data the yearly unit change in genetic improvement with, say, the genetic phenotypic approach, so with progeny testing. This is Holstein cattle, and what we obtained from 2009 to 2017 using the genomic paradigm of selection. And as you can see, the genomic approach is working. It is working very well. And so now is the reality and is the selection approach. With this, also farmers started to say, hey, why I should use genomic selection only on the male side? I like to use it also on female side. So they started to genotype female. The cost of genotyping is reducing a lot. The advantage that they can have is huge in terms of accuracy of selection, because it is equivalent of a progeny testing of 20 daughters, something that a female could never have. But so they started the genotyping, and Guinan et al. Also is showing how the genotyping in female population is increasing. So that was the first application. But we were discussing with farmers, and we started to say, hey, but why should I use only genomic and genotyping, S&P genotyping only for genomic EBV? I can take some more out of that. What are the topics? So we took advantage of the operational group project funding from Europe, the EAAP Agri resource, and we came up with a project that originates, where the goal originates from, and bring together research, farmers and farmers association and industry, AI industry. So the goal of this project was to use genotyping, to apply genomic, and to develop new process developed based on farmer needs. So we got this project funded, and we worked together with 7 farmers. We genotyped with the 100 GGP of Neogen 6,000 individuals in these 7 farms. And something that was very interesting for us was also to have the longitudinal phenotypes from sensor in farms from each cow. 2 of them had automatic milking systems, so milking robots. We have more than one for sure. All the times that they enter in the milking robots, we have a recording of all the data that they are collected. And so we have a large number of also phenotypic information. The Genotyping project was the beginning of this coupling of longitudinal data with genotyping, and now we are in the agri-tech, and that is a new funded project that is still running. And the central in this kind of approach was that we had a large number of animals with phenotype and genotypes, and especially we were working in farms where we know everything of these animals. So in somehow we can take out the uncertainty that you can have when you work on field data on a very large number of herds. Down are some publications that we have realized with this data. So what the farmers were telling us, okay, if we have genomic information, we can deal with genomic variability, how much variability there is in our farm, one respect to the others. The fact that I'm choosing one sire or another one group of females that are related one each other. And also in breeding, dealing with the estimation of inbreeding using genomic data allow to manage the animals that one has in the farms much efficiently. And then they wanted to have also a picture of monogenic loci that were affecting production traits like kappa-casein, beta-casein, beta-lactoglobulin, and this kind of loci. And then fertility haplotypes or autosomal recessive disease. So the goal was to genotype all the animals that they had in that farm, and we were able to do many things to innovate the management that they were doing. Beside the fact that they had from the association the genomic EBV, and that their selection of females was improving a lot. One farmer, the one that we are still working, said that genotyping was bringing a huge advantage for him. So he's willing also to keep continuing beside the research project. One of the things that they were also, that I didn't mention here, is that all the anaglyphic information were corrected, where errors were still in the pedigree. So the number of advantage is enormous. Here is one example on genomic inbreeding, and this genomic inbreeding is calculated as the ratio between the run of homozygosity, total length of run of homozygosity, over the length of the genome used to identify this run, and using the run of homozygosity of at least 60 megabyte. That is, say, picturing the inbreeding occurring because inbreeding mating in the last 3 generations. So you see, we can spot the individuals that have a large inbreeding, a large genomic inbreeding, and so discuss with the farmer and pinpoint to the farmers which are these individuals. So there is a one-to-one management of these individuals, and is based not on an estimate with pedigree information, but the real genomic inbreeding. I don't want to spend other words with this project. One other outcome that we had with the use of these longitudinal data with the genomic inbreeding was to use them in an approach of machine learning in genomic prediction and phenomic forecasting. This work was done by a student of the Polytechnic of Milan. He's a informatic engineering PhD now. Now he's doing a PhD in data analytics and decision science. So he was using all the longitudinal data of this farm, plus all the genotypes of these cattle, in a machine learning approach, neural network. And he was able to predict the phenotypic production, the lactation curve of cows, applying machine learning and neural networks. So he tested different approaches, and if you are interested, they are published in a paper of computers and electronic in agriculture. And the idea is to improve our use of machine learning in analyzing the longitudinal data coupled with the genotyping information. So we like to move from genomics to phenomics, and with the farmers, we're still working with them, asking them, if you have this information, if you can have an information of how much a young calf can produce in her life, is this useful? And of course it is, and we are discussing how to develop a possible tool to improve decision making for the farmers. Now, all this information went also in the new project, PNRR. It is the resilience and renovation funding that we have at national level. And also working in this, keep working in these farmers. We are working in identifying efficiency and resilience capability of these females, and also greenhouse gas emission. So we would like to also identify loci, QTL. This is the work that Andrea is in the United States with Christian Malteca. He's working with Christian in North Carolina State University. So having all the daily production in the approach that was recently published by, don't remember the name of the scientist, Poppe. So we can somehow come up with an indicator of resilience along the lactation curve. So all these peaks down the estimated, say, predicted lactation course are events that means a loss. So the more variable is the variation across this line, the more the less is the resilience of the individual. So we can estimate parameters, we can calculate indicators of resilience that we are studying about that. and then using also the data that we have on each individual cows, also try to run GWAS analysis and identify genomic regions that are related to resilience and to efficiency. That's what we are doing on Holstein. All these studies were done in farms that were Holstein cattle. So the farm were farm located in the northern flat with high technologies, and large farms like 600 milking cows. Some other work has been done on the Valdostana cattle. This is the picture that you see is the Valdostana, is a Castana, and the one in the first line here, and the red pie in the other one. We are running the project that Andrea was mentioning before, the Genova. This is a print project. Why I mention you this population? I like a lot the work we are doing in this population. Aosta Valley, it's a valley of North Italy. It's located northwest of Italy. And as you see, Italy is only partially a flat area. We have the Apennine, and the Alps are 60% of our territory, more or less, is mountain territory. So this breed is characterized by small herds. It's a dual purpose cattle. During winter, they are kept in facilities in the valley, and during summer, they go in high pasture. So they are completely different ecotype of individuals. They live in an ecosystem that is completely different of one of the Holstein. So those are the three breeds: the black pied and chestnut. This is the chestnut at the Rennes, because now they exchanged the hair book, and the Aosta red pie. The population is about 14,000 females for the Aosta red pie, and 5,600 for the Castana and black pie. Why we are interested in this population? Because this population was coming during the 15th century from Northern Europe, from the Burgundians. And then the Aosta Valley is a kind of isolated valley. So if you have the chance to drive in Aosta Valley, you will see that you have an entrance that is very narrow, and now there is a road and a highway, but this was very well, the entrance of Aosta was very well defended by a castle here. So it was basically an isolated valley. So the red pie particularly was living completely isolated with no crossing with other populations for more than 1,500 years. The only possibility of exchange was this San Bernardo pass, small San Bernardo, and the big San Bernardo. But here we have some exchange with the Ren and the chestnut, but not the red pie. So what we are interested is to analyze what kind of genetic adaptation and what peculiarity this breed is carrying with respect to other breeds. Of course, they are linked. They are a unique link with the product, which is the Fontina. And I just go very quickly. It's very fundamental that they have some economic revenue. Otherwise, they will just disappear. This is one of the part that farmers are paying attention to, is the bataille de reine. Given they are on pasture, they have this non-cruent kind of, let's say, fighting. But as you see, is that kind of things that is getting a lot of people. What is the breeding goal? The breeding goal was kind of improve a little bit the milk production with quality, a little bit fleshness. The Castana has some weight on fighting ability, But what is interesting for me is that they go to summer pasture, and farmers will never keep one of those cows that is not able to cope with summer pasture. So what we try to analyze is how the genome of these cows that was adapted to natural farming evolved with respect to the one of the Holstein. One of the structural variations that we were looking for is the copy number variant, which are structural variations related to adaptation of genome to environmental pressure. There are a lot of publications, especially related to capacity to digest starch with the amylase gene. Now I just jump this slide. What are copy number variant? Are large tract of DNA that can be deleted or duplicated, and I just jump on this. As you can see here, if we look at the variability between the three breeds, the Aosta, Red Pie, and the Castana and the Chestnut, they are completely separated, very well separated if we do a principal component analysis with the SNP's genotypes. The SNP genotypes that we obtain are always done with the 100K, so they are fully comparable with the one I show you on the Holstein. But if we get the copy number variant, structural variation in the Aosta cattle and in the Holstein, we see that the Aosta cattle do not cluster separately as before, while the Holstein is clearly separated as before. And mining the annotation that we have on genes in regions that are proper of the Aosta and of the Holstein, you see here the Venn diagram. We found that there are genes annotated that clearly show the capability of the Aosta to cope with the alpine environment, while the Holstein has duplication or deletion in different region that make her more adapted to the environment where they are found. One of the step forward is the application of genomic estimation of genomic ABV in the Valdostana. Together with another project that is the dual breeding, we have more than 10,000 cows now genotyped in the Aosta breeds, and they are keep improving. So we are aiming to have more than 13,000 this year. So Enrico, together with Francesca, Enrico is from Padova, Francesca is my group. They are developing a genomic evaluation using a single-step GBLAP in the Valdostana, and the evaluation was officially approved by the technical committee that was done in the last December. So this autochthonous and local population has now implemented the estimation of genomic EBV using the genotyping. Now, one other use that we are doing of genotyping is with another project is on goat. Capra means goat. And is run by my colleague, Professoressa Strillacci. We genotyped so far 1,600 goats out of four farms, a partner of the project. The structure is the same that I showed you before on GenoRip. We are aiming to have 2,000. Here we did some analysis of see how the genetic variability is within farm, and those are the four farms. Is principal component analysis and across farms, just to map what is the genomic variation in the population. Again, we calculated inbreeding, genomic inbreeding, and discuss with the farmers what they have in their farm and giving insight for them to map to manage their individuals and reproduction. We have run of homozygosity analysis to see where we have signature of possible selection that have been occurred in these two fog farms. And one study that was published and that was interesting is to explore the genetic variability of the PRN and P gene codons, those numbers in those goats, and we compared with the other reference in the literature, and that was interesting because we didn't have a recent analysis of this. Before to finish in 1 minute, this is the new campus where we are located now in Lodai. Is 20 kilometers southeast of Milan. I like to say that we have two bachelor courses. One especially is in breeding and welfare of companion animals. The other one is animal production. So this is livestock for food production. We have a master program in animal husbandry science and technologies. So the workshop that Andrea mentioned before that will be hosted in Milan is very interesting also for our students and for the people teaching in this course, because he's focusing exactly on companion animals. So I'm very glad for this workshop. I hope to see you guys over there. I know some of you is already have been involved. And that's all. This is my group and all the people that is allowing me to present you this work that has been done for the majority from this group of people. Thank you very much for your attention.
Thank you very much, Alessandro, and thanks Professor Bagnato. I don't see, I mean, there is something, but we don't have any questions now, but in any case, we couldn't have time because we are at 3:40, and I would love to go to the next speaker, but thanks a lot for Alex.
You're welcome, Andrea.
Very interesting presentation. I enjoyed it a lot.
Of course, beside today, if someone, I will be available for comments, you know.
Yeah, but stay tuned because until 5 o'clock at the end, we will have this general discussion, and I'm looking to have, to have you around there. Please let me remind to the people, if you want to have, if you have question for Professor Bagnato, for the next two speakers, please write down, and we will read those questions in the last 20 minutes of this webinar. And thanks also for reminding Alex for the workshop on companion animals that I didn't say before, but Professor Tosso Leb, which is our third speaker, is also invited speaker at this interesting workshop on dogs and cats, companion animals. And now we go to the second speaker of the day. You can see from what he's doing that he's from Argentina originally. I think he's preparing a mate or something like that, Sebastián, right? But currently, Sebastián Demyda Peyrás, he works at the University of Córdoba, where he's assistant professor at the Neogen group in the Veterinary School at University of Córdoba, Spain. Sebastián is a research focus on horse genomics, trying to understand the mechanism associated to fertility and sperm quality. He also studied the effect of inbreeding, productive and reproductive traits in horses and cattle. As a researcher, Sebastián participated in several international initiatives focused in horse improvement using genomics and detection of disease in horse and cattle, with emphasis in chromosomopathies. Currently, he is developing genomic markers associated to performance in Polo and Peruano de Paso breeds. The presentation from Sebastián today is about integrating genomics in equine breeding, current advances and future perspective. So we move from the cattle and goats of Professor Bagnato, we go to Professor Demyda and Peyrás, discussing now about genomics applied to the equine species. Please, Sebastián. I think it's frozen.
It's frozen, Andrea.
It's frozen, so I don't know, we can wait some time. So I don't want to ask Tosso to anticipate the speech because would not be... Okay, here it is.
I don't know what happened.
Yes, okay. You were frozen before. Please, I introduced you the best I could. So please go ahead and make your presentation now, Sebastián. Thanks.
So are you seeing my screen, right?
Yes, we can see. Thanks.
Thank you. Well, first of all, thank you for inviting me. It's a real pleasure. I'm not a common user of attendant of EAPs, but I hope that in the future I can go more. And the idea is to give a little glimpse about herd genomics, how herd genomics is in the currently is doing, right? See, okay. So yeah, the outline is where are we? Sources of genomic information, current uses in genomics, the talk aims to be applied, and what about the future a little bit, okay? So genomics is using information, thanks the previous speaker, Alex, that explained a lot of things that I will explain now. So I have half of my work done. But it is important to understand that genomics is not developed the same in all the species, okay? So if we think about dairy cows and pigs, probably horse genetics is so big, this big as us. We are far behind the other species by different motives, by different causes. But I will try to convince you that in the future, this small horse in the lower part of the screen will be much higher or much taller. Okay, yeah. So genomic information sources, as Alice said, there are three basically that are microsatellites, SNPs, and NGS. In horses, because we are a little bit far behind. STR and microsatellites, STRs, are still using, but mostly for parentage. I will say only for parentage, and this is because it's an old technology that produces a lot of problems and biased results, mostly in inbreeding when I use it for other aims that parent test. In parentage goes well, goes perfect, but for the rest of the things is quite more difficult, right? And also it's not informative at all or not accurate to genomic level, right? Okay, sorry, my computer is being silly. Okay, and why still is in use in horses? Basically because several breeders associations are like old-fashioned, and it works for changes, right? And also for economics perspective. So migrate from one technology to a new technology for two parentage needs money because you need to adaptate the old results to the new technology. So at the moment, I will say that only microsatellites are being only used in parentage testing. I'm sorry. On the contrary, we have SNPs. SNPs are much more informative and much more common, and is nowadays the technology of election. It is being used in most of the species. It's increasingly used in horses. It's highly informative at individual level. You can get a lot more information of SNPs that using microsatellites, and the cost-benefit quality is great in terms of information. Some problems, yes, of course. Low flexibility, because you can't do array you can't change the arrays, and but the data sets are relatively easy to analyze. Okay. Nowadays, the problem in horses is there are few arrays of SNPs available. There is only one of high density, the axion of Thermo. There are 2 MD arrays available using Illumina technology, and there is a array recently developed of medium density, 90K about, with thermo technology. I think that it's not yet publicly available, but it will be soon. But in general, the most used is the Neogen GGP 70K, right? Is the most common use array right now for horse genomics. Lastly, NGS. NGS is the technology of the future, probably, but this is still because it has a lot of improvements with respect to SNPs, but this is still not available for breeding. Maybe for research, it's a good technology, but it's not good for breeding. Basically, because the price is still prohibitive, and it requires a lot of computational capacity that is not available among breeders associations or kind of. Okay, so we focus this speak in this talk in SNPs, right? Okay. So for parentage testing, the ISAG International Society of Animal Genetics recently developed a new panel to start to do parentage testing also by SNPs. Okay, this is a huge step forward because it will be improve or increase the adaptation of the technology, right? Comparison tests will be available in 2026, and most of the I will say modern breeders associations aims to migrate because they have a lot of information, additional information using the same analysis that for parentage testing. The problem nowadays is who pays the migration, basically, and SNPs are a little bit too expensive for some production realities, like South America, for example. Okay, but in the future it will be. It's not if it the technology will change. It's more like when. Okay. Populational studies. SNPs are great to differentiate populations between populations. Just a biological sample here, and you can get a glimpse of the genomic makeup of any individual, and by comparison with different breeds, you easily can differentiate, right? There is a lot of studies and papers published on differentiation between breeds. This is, for example, in Arabs, different kind of Arabs, different type of Arabs horses, like endurance or racing or show, and you can see that there are differences, right? Differences are not so big like with thoroughbreds, that is another breed, but you can differentiate within a breed with individuals with different capacities, which is really important in the multi-proposite breeds, right? There are different technologies to do that. This is probably the most comprehensive. It's a mixture. And as you can see, you can differentiate very easily the thoroughbreds, blue in the right, but using more finer analysis, you can also differentiate very easily between the different populations of Arabs, okay? So this is available. In our group, we work with Cartujano horses. It's a very old lineage of the pura raza español horse. Cartujanos are closely breeding like 500 years, maybe. At least they say that. And you can see that within the same breed, you can see very easily there are two populations, right? And the red ones are mixed, are a mixture, individual, a mixture between the two breeds. So it's quite clear how, using genomics, you can say that this is a Carthusian and this is not a Carthusian, which is quite important in the closed breed associations, closed stud book associations, because if they need, they can certify to some extent the origin of the horse, the lineage of the horse, okay? This was made with our long-time collaboration with the Pura Raza Español Breeds Association here in Spain. In polo ponies in Argentina is, I think, the only thing that Argentinian people is the best in the world, besides football, but this is more in discussion. But in polo, there is no discussion. Argentinian people create a breed, right? By selecting thoroughbreds, basically, and mixing with individuals with certain mental characteristics, right? If you see, the blue ones are the thoroughbreds, the red ones are the polos, and you see there is not so much differentiation, right? But after 3 generations, you can see that it is differences in the genomic makeup. So in 3 generations, you can already see some differences between the different lineages on horses, right? So genomics is a very powerful tool. The next one is inbreeding. Inbreeding is, Alex talked about it. Inbreeding is a particularly important problem in monarchies. Here in Spain, there is a big problem. There was a big problem, right? He killed a European royal dynasty. But horses is also a big problem, right? In general, inbreeding horses is a problem, right? Pedigree-based inbreeding is very reliable when the pedigree is robust, right? But if you have breeds like polo, which is a mix of different ones, right? And you take the whole pedigree stud book, you probably get this one. All the horses are not inbreeding because a couple of generations ago, we have no more data, right? It's 4 years, it's 3 generations, 4 generations, right? So when you get inbreeding, genomic inbreeding, the precision is skyrocketed, right? It's quite more precise, and you can manage quite better the inbreeding because you can do genomic related matrix, and in base of that, you can select or predict the crosses that will maintain or reduce the inbreeding, right? So this is also a tool really available, and that is being used, for example, in Argentina in polo. And also, you can, using molecular inbreeding, you can see the effect of inbreeding more topological, right? It's not the same that you have a lot of homogeneity in the chromosome 1 that in the chromosome 3. In this picture, you see different chromosomes, and the inbreeding level is totally different, right? It's totally different, even within the same breed. So using this approach, you can get a fine-tuning of inbreeding influence and associate to traits, right? We did that also in Spanish pure breed, in Pura Raza Español, associating different kind of inbreeding with fertility, and the results were very clear, right? This is the variability in inbreeding at chromosome level, and you can see that in this part, this peak of inbreeding chromosome 7 affects, but this one in chromosome 8 that is almost the same extent does not affect at all the reproduction. So it is not the same. It's more fine-tuning, and with that, you can get more reliable conclusions, right? Genomic and monogenic diseases. This is probably the most used approach in horses. You have a lot of diseases in which the genetic makeup or or basis was cracked, and when you crack the genetic basis of a disease or a trait, you can produce easily a test. And most of the tests can be used by SNPs, right? So in the same array, you maybe have 100 or 150, markers associated to different traits, right? In this case, I don't know if somebody see this before, but there is a naked foal that falls with no hair in a very old Russian breed. There is a test for it. It's just a test that can predict, right? Polygenic diseases, that is a totally different, right? Because you don't get one specific marker for this disease. In this case, it's Peruano de Paso, and it's a problem in the hoof ligament, right? They become more lax, and the horses have problems, right? A couple of years ago, the people from Wisconsin, I think, they do a GWAS and develop see a very big signal here and develop a test, and that's it. You have an algorithm. In this one in particular, it's under patent, but you have an algorithm that can predict the outcome of a mating between two individuals in terms of how lax will be the tendons of the horse at adulthood, right? This is also available, and it's being used in Peruano de Paso horses. The same for fracture risk in thoroughbreds. There are markers associated, these three in particular, associated to increased risk of fracture, spontaneous fracture in thoroughbreds. Again, this was made 10 years ago and is still in use. In selection of thoroughbreds, right? Then in horses, coat color is quite important in several breeds. For example, in Criollo Argentino breeds, different coats have different price. Horses with different coats have different price, and most of them are monogenic. So basically, you can get markers or the genomic marker for any breed that you want, any coat color that you want, using genomics, right? In this case, it's a Pinto, it's a monogenic loci that gives this coat color, or the Appaloosa. In this case, need to be heterozygous, I think. I'm not expert, but right. In the presentation, there is a Etalon. This is a company in USA, has a nice simulator. This is a simulator that you can make your horse, all right? So you can predict your code, the code of your horse also, right? Behavioral traits. We use in polo. There are some behavioral traits that are associated to genetics in a little bit. It's not a monogenic trait, but they have like a how I say like a tendency to have some kind of behavior. And in polo is being used right now for select horses. Like accessory traits, it's not the most important, but it's being used. All right. The most important problem probably around the world, that's the work of Emery Hill in the type of muscle fiber, right? Is the speed gene, known as the speed gene, that can predict very accurately, based on one single loci or a couple of locis, if your horse has a muscle makeup more for short distances or long distances, right? So this is also under patent in I think it's worldwide, but you can send a sample of hair of your horse and get a very reliable approximation if your horse will be better running short distances, they call sprinters, or if the horse is better for long distances, that they call stayers, I think, right? And this is a monogenic trait, so you can predict matings. You want some kind of course, a sprinter, okay, you can make it based on the parentage of the genotype of the parents, right? The same we are using for the age of the horse. There are 4 loci that explain a lot of the height of the horse. In polo ponies, for example, we need small horses that we select using that ones to get small horses, right? And also there are some markers, polygenic markers associated to morphological traits. There are less precise, obviously, because it's not so easy, but there are several studies that you can see that different markers are associated to different type of morphological traits. Elbow joint, stifle joint, feet joint of the hind limb, carpal joint, and you have the marker, right? Functional metabolic syndrome, there are markers associated also to metabolic syndrome in which you can predict how possible is that your horse get a metabolic syndrome when it's under is restrained, okay? It's not free ranging. And also the functional ability. There are 5 types of quarter horses, and there is a study in which the people from, I think, Minnesota associated the genomic makeup to the different ability. So you can predict a little bit what is the best ability to your horse, okay? The same with the reproduction. The same with reproduction. We did this study a couple of years ago with Nora, one of our superstar postdocs. And we found several markers that explain a little bit because it's reproduction fertility of mares. So some markers make the mares more fertile than others, right? This work was also made with the collaboration of the ANSE. And also we have a long collaboration with the ANSE with chromosomal abnormalities. Horses has a lot of chromosomal abnormalities. And using genomics, we develop a methodology that can easily detect most of the chromosomic problems in horses. We did that, yeah, like 10 years ago, no, not 10, but 8 years ago. And now this is used routinely in the Purebred Spanish Association to detect very early any horse with a chromosomal problem and to manage them better, right? Finally, the future. Well, the future is following the steps of the other species, mostly, right? This is one of the things that I think that will be possible soon, really soon, that is embryo biopsies. There is a very explosion of ICSI technologies in horses. And if you have ICSI, you can get biopsies. And if you get biopsies, you can get genotyping. So soon, you can get your embryo with a genomic evaluation, right? Before decide if you keep it or not. The other thing is future, a little bit of reality. There are several breeds. I will talk a little bit about the one that I know, that is Pura Raza Español horse. Going to genomic evaluations, right? As Alex said, that a little bit about cows that is fully developed in several breeds. In horses, is still like in doing baby steps, right? I think one of the most robust breeding programs without genomics of horses is the Pura Raza Español breeding program, right? But in this case, you have the full sibling problem. The full sibling problem is that, as you can see, this is our Messi, you know Messi, and the two guys in the left are the brothers of Messi. So you can see that only one is really good performing, but when they are adults, right? So you need to wait a lot, right? So using genomics, the idea is that you can reduce the uncertainty, sorry, because you get like genomic evaluation earlier, right? Reducing the interval between generations and get a more increased breeding improvement, right? So basically, the idea is using the same models, but changing the related matrix using pedigree by a genomic matrix that is much better to see differences between full siblings, right? If you see a picture of my two sisters, you see big difference. And I think that we have the same parts, right? This is, I think, last month is the first one in the pipeline, and with the people of the ANSE, comparing the genetic accuracy. How accurate are you modeling the traits between pedigree-based breeding programs and genomic breeding programs, right? As you can see, this is the right is the genomic, and the left one is the pedigree-based, and the reliabilities are quite similar, right? The reliabilities doesn't change a lot. But the accuracy in different traits, all of these are morphological traits, is quite more accurate, right? And we are using a little bit of genomic information. This one, there are only 4,000 horses genotypes, right? When we have 10 times that, the accuracy jump will be much higher, right? But using a little bit of genomic information, we get 10% increase in accuracy in some traits, which is a lot, right? It's raining here. This is also work in progress. I think in a couple of years, genomic will be integrated in several breeding programs, and prediction, of course, progeny will be more. And also because you can select very early, this is yearlings of Polo, you can select that one based on the genomic, and this one that looks very nice is not the very nice one, because we will not perform. In this case, she will not perform, okay? And finally, a little bit of genomic and clones. This is also another program that we are developing. I think that probably almost of you know that cloning horses, now there are like 1,000 cloned horses in Argentina, which is quite insane, but it is. So clones have different mitochondrial makeup by the procedure, right? They use a nucleus of the founder, but using all sides of different mares, donor of mares. So mitochondria are different between clones and founders, right? There are several works saying that mitochondrial profile can affect horse performance, right? So using genomics, because mitochondria is the powerhouse of the cell, right? So using genomics, we can see the genomic makeup of the mitochondria of the horse. And you can see this one is the original. Some clones are very similar, and others are totally different, and others are even more different. So there is an ongoing project that will have some result probably the next year, trying to see if that affects. Because we have 1,000 clones in Argentina, but 10% is playing, right? Okay, that's it. A little bit of closure. Genomic techniques are here in horses. Several tools, mostly marker assist selection, are being used. there is a lack of knowledge in several breeds that have zero knowledge about the advantages of using genomics, particularly in South America, which we have a lot of horses. New tools are currently being developed every day, like every day. Code color is a new one every 2 months. As Alex said, genomics needs help from phenomics. So the problem here now is increase the using of genomics and improve our phenomics techniques, right? So in the future, we hope that the horse is bigger a little bit and is coming at full gallop to increase the difference between species. And it will, I don't know when, probably steadily, but slowly but steadily. But yeah, there are a lot of people working. There are beautiful works made around the world, and it's just we need to wait and see, right? So always keep calm. Ask a geneticist. This is the geneticist. This is our group. People from Spain, the big bosses here in Spain. People from Argentina. Horses from Argentina, that kind. Funding is from the agencia in Spain, basically. We have a social network if you want to hear something else of horse genomics. And I think that's it. Thank you for inviting me again. And I take pictures with horses, with people. Those two are the most important stallions in polo nowadays, the Guitarrero and the El Padrino. So I'm very happy with those pictures.
Well, thank you, Sebastián. Thanks for this presentation that take us to the world of horses, and also another kind like strange environment for us as European, like polos, which is, is very common and popular in Argentina. In Europe is much less. But it's so fascinating to hear about that. We will ask, at least I have a couple of questions, but I will keep them for the last moment of this webinar. I remind again everyone, if you have questions to Sebastián, to Professor Alessandro Bagnato, and to the next speaker, Tosso Lab, please write down in the chat, and then we will read them. Now it's time. Thank you, Sebastián, again.
Yeah.
And now it's time for our last speaker. Well, I love to say the usual words, the last but not least, something like that, but also is a very famous scientist worldwide. He's working in the University of Bern, where he's professor of veterinary genetics. His TOSO research focuses mainly on the identification of causal genetics variants for inherited traits in domestic animals. His group, the group of TOSO, has found more than 100 causal variants for hereditary disease in dogs, cats, and other companion animals. TOSO presentation title today is a high-density genotyping solution facilitate the identification of disease-causing variants in companion animals. So thank you, Tosso. You have 30 minutes like everyone else, so I expect you finish by 4:45, more or less. Thanks.
Thank you very much, Andrea. Thank you very much to the organizers for inviting me to give this presentation. And I thank my previous two speakers who introduced the topic a lot. So this one is focused really on discovery of disease-causing variants in companion animals. In cats and dogs, we have many breeds, but the breeds typically have very small populations. There's a lot of inbreeding going on. And there is a necessity to monitor especially recessive diseases, recessive defects, to develop genetic tests. And my research group is involved in that activity. And I'm going to talk a little bit how we identify disease-causing variants to enable the development of genetic testing for companion animal breeding. I will give you a few examples. and they all involve whole genome sequencing, which has become pretty much a standard technique for us. Again, we have small numbers, not like you heard in the first presentation where thousands of cattle are genotyped. Typically, we do not have much a priori genotype information, and very often we have single cases of dogs or cats that show a disease. So I will briefly talk about single case whole genome sequencing, and then I will give examples of how we use positional information, either linkage or genome-wide association studies. And I will very briefly touch the challenges with structural variants. So the goal of our research is we have a companion animal, a dog. The rest of my talk will be focused on dogs with a given phenotype. And the goal of the research is to identify the causal genetic variant. And you could differentiate. We need to identify the right gene which is involved, and then within the gene we need to find the causal variant. And to identify the right gene, we have two basic approaches. One is functional candidate genes for specific phenotypes, and the other is positional approaches that are hypothesis-free and basically work for every disease and every trait that we study. And on the variant discovery side, in the past, we used a lot of targeted sequencing by Sanger sequencing technologies. We rarely use this anymore. Nowadays, we focus mostly on whole genome sequencing, which has become so convenient and cheap and efficient. When we come to the two approaches to identify genes, we have functional candidate genes, and this is a very fast process. You have to know the genes that have been associated to specific phenotypes. So it can be very fast, but requires some expert knowledge, or you need to know the right databases. You need to know a little bit on basic biology, biochemistry, and physiology. If you write grants, the good thing is it's a hypothesis-driven method, which granting agencies like a lot. However, I would say it's really a guessing game, and it tends to be quite error-prone. And we see many traits where we have either no candidate gene, or we have a very large number of candidate genes, and then the information is maybe not so super helpful. For example, if you have neurological things in humans, developmental delay when kids are not developing in the normal way, there's now a database that lists more than 2,000 candidate genes, and this can be very challenging. On the other side, the positional approaches are really hypothesis-free. The same methodology works for any trait. You don't need basic knowledge on biochemistry or physiology, very convenient. The disadvantage is these positional approaches do not work with a single case. You need a handful of cases and a handful of controls to make them efficient. And I will show you some examples. But for monogenic traits, and this is my main line of research, you don't need hundreds of animals. It can be fairly small numbers. And then whole genome sequencing, just a very brief recapitulation. Mammalian genomes are about 3 billion bases and have 20,000 protein coding genes. There's no big differences between different mammalian species. What we do routinely, we put a lot of emphasis on preparing the best quality libraries, which means they have to be PCR-free without amplification steps to have very even coverage, even representation of the genome. This means we need reasonable quality DNA, but I mean, it's nothing very fancy. But yeah, the DNA has to be of reasonable quality. And typically we sequence at 20x coverage, somewhere between 20x and 30x coverage. That is sufficient, in our opinion, to have a very good sensitivity to detect germline genetic variants. We produce FASTQ files, and those are aligned to a reference genome. Variants need to be called. And then very important, we need to annotate those variants, and the quality of genome annotation that varies a little bit between species and also between agencies. So we prefer NCBI annotations over ensemble annotations. Yeah, this is very important, that the quality of the annotation is high. Otherwise, you get many false positive potential candidate variants that you then need to filter out in the end. So you can imagine if you sequence a given animal, we will find millions of variants. And how do we find the variant that's causing the hereditary disease that we are studying? Again, we employ positional approaches where we need to know where to look in the genome. We employ functional candidate genes. We search for private variants. So we compare the genome of our diseased animal to many other genomes and look for variants that are exclusively present in the diseased animal. And we prioritize protein changing variants. So nonsense, missense, splice site, frame shift, and so on. In the annotation, we use a software called SNP-EFF, and that classifies the variants according to impact. So the high variants are those that are predicted to lead to a complete loss of function of the gene. Most important classes are nonsense, splice site, and frameshift variants. And the moderate ones are the missense variants. And low and modifier, these are not necessarily predicted to change an encoded protein, and normally we ignore them. And now to give you examples, this is a dog that was referred to us, a Rottweiler with footbed hyperkeratosis. So you see these fissures in the paw pads. That's quite painful to the dog because this can get infected. In histology, you see that the epidermis is massively thickened. And to find the variant that's causing this, we sequence the genome of this dog. This case a little bit higher coverage than what we do normally. We compared the variants to the genome sequences from 655 other dogs that we had sequenced in previous projects and looked for variants that were exclusively present in this affected Rottweiler dog. And when you look at the numbers in dogs, we typically find when we sequence a particular dog genome, we find roughly 6 million variants compared to the reference genome. 3 million of those are homozygous and 3 million are heterozygous. This is an average number. And again, when we do this search for private variants, the numbers come down quite a bit. And when we focus exclusively on private and protein changing variants, in this case, we ended up with 29 variants for homozygous, 25 heterozygous. And this is then a number at which we look visually. We export these variants in an Excel file. These are the 4 homozygous variants, and they affect 4 different genes. We do searches on the internet. Is anything known about the function of these genes? And the one that's highlighted here in yellow affects a gene that has a known function in skin. And this led us to the conclusion. So again, literature search. There are 32 functional candidate genes for similar phenotypes in humans. One of them is the desmoglein gene. And then out of the 29 variants, only 1 remains. And that turned out to be the causal variant in this case. So in this case, a frameshift variant that really destroys the gene. And it was quite interesting to see in this case, the dog was referred to us with this footbed hyperkeratosis. And we found this gene, which is known in humans, and it causes a phenotype in humans that also involves changes to the skin in the hands and on the sole of the feet, which is the homologous phenotype to the footbed hyperkeratosis. But the human patients, in addition, have very severe allergies because they have a skin barrier defect. So we went back to the veterinarian and asked them, we think we know what's going on in this dog, but is this dog allergic? And the veterinarian said, yeah, the dog developed very severe allergies, but we thought they were unrelated to the footpad problem. So in this case, the genetic analysis also helped to understand the disease better And yeah, to realize that the allergies are not there by chance, it's part of the phenotype in this dog. Another example, same line of research, a dog was presented to us with Ehlers-Danlos syndrome, so that's hyperelastic skin that tears very easily, a very well-known phenotype in dermatology, humans and animals alike. And currently there are 20 candidate genes for Ehlers-Danlos syndrome known, and we sequenced this dog. And this is the pipeline. And again, same procedure. When we did all these filtering steps, only a single variant was left and led us to the finding that this is a variant in collagen 5 alpha 2 called 5A2, a known candidate gene for Ehlers-Danlos syndrome. So when you have single cases, it's sometimes, sometimes it's not possible to solve them. You have to have a little bit of luck, and it's especially challenging when genes are involved that are not clear candidate genes beforehand. And it makes the chances of success are much better if we have several affected animals and families. And when we talk about dogs, dogs are born in litters, and these families are just wonderful for genetic analysis. And what we can do if we have a complete litter with both parents, we can do linkage analysis. And in this example, these are golden retrievers with ichthyosis. So they form scales on their skin. It's not a very severe disease, but still it is a disease, and owners should know about it. There is a very common form of ichthyosis in golden retrievers that is well known. And this refers to a new form that appeared in North America that tested negative for the previously known form of ichthyosis in golden retrievers. And the breeders were very diligent and careful to compile pedigree records, and they compiled a very large family with several affected dogs. And in the end, we were provided with samples from 44 dogs that came from 7 different litters. And then we need genotype information, and in the last years, we very routinely use the Illumina K9 HD microarray. It has 220,000 markers, and Neogen provided wonderful genotyping services to us. We prune the data, so we remove non-informative markers and markers that are not genotyped in all of the samples. And then we perform linkage analysis with the Merlin software. And the output are lot score curves for all chromosomes. It is just a subset of the dog chromosomes, and you see these curves, and the expected result, and in this case, it really worked like a textbook case. We have only one chromosome where we have a meaningful region with positive lot scores. That's here on chromosome 23, where the maximum lot score is 2.75. So that would not be sufficient to claim proven linkage, but it's the only region in the genome where we have positive lot scores. And this is clearly the region to look at for the causal variant. What we can complement the linkage analysis with homozygosity mapping. That is a fine mapping technique where we look for homozygous shared haplotypes in these individuals. And when you remember the pedigree, we had 14 cases. So we looked in these 14 cases for shared ones of homozygosity. And this picture shows where we have linkage in yellow and where we have homozygous regions in red. And there's only one region where the two things overlap. And this gives us a critical interval where we have to look of 12.7 megabases, or 0.5% of the dog genome. So we excluded 99.5% of the variants in the whole genome sequencing experiment. And when we look at the numbers, again, 3 million homozygous variants, private roughly 1,000, but in the critical interval we come down to 32. And now we don't need to look at functional candidate genes, or that is no longer really important. The next step would be that we look which of these 32 variants are predicted to affect a gene. And again, in this case, we were quite lucky. There was really only one that was predicted to change a protein, and another deletion in a gene called ABHD5, which is actually a candidate gene for ichthyosis, but it was not one that we had on our primary list of candidate genes. The other positional technique that we can employ is genome-wide association study. And this was already nicely shown in the two previous talks. Just to remind you, if we look for monogenic traits, the sample numbers required for successful GWAS are quite small. If it's really a monogenic trait, we can get away with very few samples. And the example that I want to show is exfoliative cutaneous lupus erythematosus, ECLE. That's an autoimmune disease. Normally, autoimmune diseases are not assumed to be monogenic, but in these three hunting dog breeds, the ECLE is a monogenic disease, and this has been studied intensively and known for decades that this exists, and hundreds of dogs have succumbed to this disease, which is quite severe and debilitating. And we were provided with samples, fortunately, by Maggie Casal and colleagues from Pennsylvania, and performed a genome-wide association study, 14 cases, 29 controls, and you see it's a very clear signal on chromosome 18. Again, we complement this with homozygosity mapping. I won't go into details, that's all clear to you. So these 14 cases share a homozygous segment on chromosome 18. And when we look at the shared homozygous segment, it's less than 500 kilobases. So we have, in this case, a super precise mapping, only a very, very small piece of the genome. Still contains 26 genes, which are listed here. And when we look at the sequencing results, so in this case, we sequenced 2 cases. We look for shared variants. In this very small critical interval, we remained, or only 2 variants remained, and only one of them is protein changing and was a missense variant in a gene called UNC93B1, which really did not have a known role in autoimmune disease at the time of our research. During our research, a few months before we published this, the function of this gene became clear, and there were 2 publications in Nature about its function in mice. But it was very reassuring to us that we can find genes that are not clear candidate genes for a disease if we have good positional information and this approach. And by now, I think in the last year, also a series of human patients with autoimmune disease caused by genetic variants in UNC93B1 has been published. To sum up what we have in terms of positional mapping techniques, we have genome-wide association study that requires unrelated animals in the case control design. We have linkage analysis, which requires complete families, very convenient in dogs or companion animals where we have litters. We can complement this with homozygosity mapping if we are talking recessive diseases. Normally, it's always after inbreeding events, so the affected animals are expected to share the same causal variant on the maternal and the paternal allele, and then we can do homozygosity mapping. And if it should be a dominant disease, we could instead do haplotype analysis, which I have not presented in detail, but which works along similar lines. A final last example to show that sometimes it's not quite as easy. This is a dog, Haley, presented with another skin disease, quite characteristic, linear lesions on the skin. Haley is female. So here are some pictures of the dog. We did our standard approach. Because of the spatial distribution of the lesions, we had like an idea that this could be an X chromosomal disease in a heterozygous female. And we ended up with a number of variants on the X chromosome. However, none of them was protein changing. And now I have to remind you, in Illumina sequencing, short read sequencing, this is very sensitive to detect single nucleotide variants and small insertions, deletions, but it is not useful at all to detect structural variants. So the software tools, the normal variant callers, can only call small variants. And according to our experience, it's impossible, or at least near to impossible, to reliably call structural variants from Illumina short read sequencing data. So yeah, in our hands, we use the automated pipelines only to detect small variants. And when there is no small variant, then yeah, of course, you start to wonder, is there a larger variant? And then again, in our hands, we cannot rely on fully automated genome-wide searches. Then we need to know where to look. We need to have a candidate gene or some positional information. Unfortunately, for this disease, there is a clear candidate gene, which is called NSDHL. And when we look particularly at this region in IGD, where you can see the raw data, then it becomes very clear that there is a deletion, actually a quite large deletion, 120 kilobases, that includes the entire NSDHL gene, and that explains this phenotype. So to summarize, what I presented today are some techniques that are useful in the genetics and genomics of companion animals. When there is already a genetic test available, you can use that genetic test to determine the genotype of your dog that you may want to use for breeding. But that's only useful if the genetic test is already available. And we see really many new hereditary diseases popping up. And if you need to search for new variants that are not known beforehand, there's targeted sequencing of candidate genes, which is relatively fast, but it's very difficult to guess the right candidate gene for many traits. And variant detection sensitivity for structural variants with targeted sequencing is really low. So this is an approach that we hardly do anymore. Our standard approaches really involves whole genome sequencing, either of the single case or even better, the single case and both parents. That's even more powerful. Yeah, has a very good efficiency. However, in our hand, takes several months because the computing to search for private variants within a cohort of 1,000 or more dogs is computationally quite intense. And our high performance computing cluster is not super powerful. So we lose a lot of time during the calculations. And ideally, if there is more than one case available, we very much like to complement the sequencing data with positional information and perform either linkage analysis or genome-wide association studies. With that, I'm at the end of my talk. I would like to thank all the researchers who did the work, the collaborators who referred samples to us, and also Neogen, who really helped us a lot getting very robust genotyping information that really helps in our research. And yeah, without that, we wouldn't have been where we are at the moment. So thank you very much to the sponsor, Neogen. And thank you for your attention. I'm happy to answer questions.
Well, thank you very much, Tosso. And from the bovine and the goat to the equine, now we go to the dogs. So we cover many species of domestic animals. And also thank you for recognizing the support of our sponsors. So today's webinar, which is Neogen. I understood this is practically for all of the three speakers. You have got support also from Neogen. You work with them. And I invite again people to ask questions. There are some questions going on around. In any case, as every good chair, I will have some questions myself. But it was not so difficult because all presentation today were so interesting for anyone, especially if someone has a genetic background. I will go through with the first question raised by a participant. Will artificial intelligence and machine learning accelerate even more the progress of genomics? And what new areas will be developed in most farming breeds? Who wants to answer to that? Maybe Alessandro?
Answer is a big word.
Well, comment, let's say this.
Yeah. Who knows? Who knows? No, my experience is that artificial intelligence is running very fast. So, for example, we are able to generate pipelines for data analysis recorded in farms. So we can ask to generative AI, for example, to take advantage of the longitudinal data collected in farms and available at farmers' ownership or whatever it is collected by farmers. If they also have genotype, also to provide genotype and ask to do analysis that we would like to have as a result. For example, genetic evaluation, genomic evaluation. I tried myself and it worked. So if you ask to generative AI, please, can you generate genomic EBV given that you have phenotypes recorded by milking robots plus genotype using this linear model, and that's it. It came up with genomic estimation of relationship according to this, and that. So this is what we can do today, piece by piece. And as far as my discussion with people that is using more heavily generative AI, they told me that probably in a couple of years we will be able to generate very complex pipeline for data analysis. As far as I know, and then we can ask, to provide a suggestion for management of individuals. As far as I know, there is people that is actually is a project of is not in RAI, is the Polytechnic of Antibes. They are developing research project to analyze all the databases that are available on NCBI or Ensemble on gene sequence, RNA, and use this metadata in an artificial intelligence context. That's more on research side. So myself, I'm trying with the people working with me. We have a PhD student that just started that will come in Zurich for the meeting, Andrea, and her PhD program is focused on machine learning and generative AI applied to animal breeding and genetics. So I think that in a couple of years we will have available something that today we cannot even imagine. Teaching to students every year, I have something that I was not able to teach the past year.
That makes sense. Science is also always development. No, I don't understand people teach the same lectures for decades.
No, but it's running very fast.
And now it's running fast, and it's difficult to cope with this speed. But I agree with you that is a lot is ahead of us, and we will see. I mean, I think about the AlphaFold experience examples. Everyone knows about AlphaFold, making the three-dimensional structure of protein, and it was already, if I'm not wrong, a couple of years ago, and now how can go so fast this development. Any comment from Tosso and Sebastián on this question?
I don't feel qualified to answer because I'm so bad at this, but I also expect very fast developments.
I guess, Tosso, I'm also very, very bad, but I try to get to show that I know a lot, which is not true. No, but that's okay. Yeah, thank you very much, Alex. This was a good answer. I have a question to Tosso now specifically, and according to you, what is the main cause of hereditary disease? Is it inbreeding? Well,
inbreeding definitely contributes to the spread or to the increase in prevalence in hereditary diseases. I mean, the cause are mutation events. Mutations happen spontaneously and they happen all the time. But with inbreeding, you get the chance that a deleterious allele becomes homozygous, identical by descent, if the animal is inbred. So that's a factor. And if you have closed breeds, closed populations, you should monitor your inbreeding, and you should definitely monitor for recessive diseases, because you have to expect that they will appear at some point. But it's not only the inbreeding. I would say the inbreeding can be managed, especially with genomic tools, as has been shown during today's talks. So with the perspective from companion animals, but it's also true for livestock, we have this super intense use of especially popular sires. So males that have hundreds or thousands of offspring, and a single individual sires 10% of the entire population in extreme cases. And I would expect that every dog, every cat, every human, every outbred animal, what we have in our typical livestock and companion animal population, will have one or even more recessive deleterious alleles. So one just has to acknowledge that these alleles are present. And when the breeding practice is not optimal, you run the risk of having the recessive diseases. So my recommendation to dog breeders is always manage your inbreeding and monitor for recessive diseases. Don't act as if this was unexpected. I mean, new recessive diseases will appear, but if they are closely monitored and action is immediately taken, my idea would be one affected animal or one litter with affected animals should be enough, and then research could be initiated, genetic tests could be developed, and then no further affected animals need to be born.
Thank you, Tosso. Yeah, inbreeding is an issue, and as you say, it contributes to the development of to realize of this disease. You just say something about, this in cattle, for instance. You say, there are popular sires that have thousands of offspring, and that will also cause inbreeding. I would love to go back to Professor Bagnato and about this, because this is always a big issue for those who make a genetic evaluation or genetic improvement of dairy cattle, for instance. We know that Holstein Friesian, which is the most popular dairy cattle breed, have a very few ancestors, and so we think that there is a very high level of inbreeding compared to what it should be natural for millions of animals around the world. But Alessandro was working, and he showed an example of a small breed that has been isolated for centuries in a valley in the Alps, which is the Val d'Ostana. Alex, what do you think would be the difference in breeding level on these two complete different breeds? Think about Holstein Friesian, cosmopolitan, all over the world, but few ancestors, with a small breed isolated for centuries in a valley in the Alps. And eventually, how genomic can help both situations?
Yeah, I feel like a student, Andrea.
You are like, just joking.
Joking, of course. You would not trust, but the Val d'Ostana has an effective number that is larger than the Holstein. We calculated the effective number based on the SMP with the SNP or other approach that was available. And the Val d'Ostana has a larger...
I thought about that, Alex. Also in my many years ago, I worked with Val d'Ostana. My questions were polemic, because we think about this, that the hundreds of millions of Holstein Friesian in the world, they should not have inbreeding as a small breed in the Alps.
Yeah, they clearly show that they have a bottleneck more than the Val d'Ostana because they were under selection. Based on the experience we did, actually I don't have an explanation, Andrea, but the level of inbreeding, if you calculate, is it has the ratio between the length of the run of homozygosity over the entire genome. I mean, in my vision, is less than what I would have expect. I don't have an explanation. Now, the genomic inbreeding meaning is very different from the inbreeding that we were used to approach with pedigree, because you really go to spot if an individual was under mating of related parents. So if you look at the long run of homozygosity, you see this kind of situation. And as far we are dealing with farmers, you can manage this situation much better than just using the pedigree breeding. I don't know if we can have an approach where you can gain genomic variability in the population. You should actually, with the GenoRip, we tried the approach with the EAAP Center to see if we were able to optimize the mating, comparing the variability between sires and then sires that you can use from an AI company with the variability that you have in a farm. Then for a reason external to the project, we were not able to finalize it. It was related to a person
Okay.
that you might think who she was.
Okay. Okay, thank you, Alex. And I have a question also for Sebastián. And
Was a good person. Sorry, Andrea.
I don't know, I imagine.
To let know to all the people, was a very good scientist and person. Unfortunately, was.
Yeah, and Sebastián, so you say that the genomics in horse is behind of the other species. In which way do you think this is? Why do you think it's late? Well, I have an idea, but we are not talking with our, people around that are listening to us. And what do you think that could be forecast of the use of genomics in horse breeding?
Well, the horses are like big dogs. I mean, it's if you think from a genetic perspective, they are companion animals. Mostly, I mean, the horses that are selected, I'm not saying the working horses, that they select phenotypically for, say, something. This is the first issue. The second one is that horse production is economically important, but very extensive. And when the horses that have high value in general has the same problem that Toso mentioned. I mean, there are breeds with 10% of the individuals cited by the same stallion, 10%. So one stallion is playing 10%. And that's make like companion animals and not productive animals. So the people is not looking for economic improve, more like a performance improve. And in that kind of mental frame, I think, it's my opinion, the people has like believes in their knowledge, right? It's like, you can't say to me how to breed horses. It's difficult. And also there are two additional problems, that horses are very nice species for geneticists, because generation interval is quite long. So you are taking decisions right now to get something in 10 years. Not in full breeds, okay? But in the rest of the breeds, polos start to play at 7 and get the peak of to 10 years. And in jump or dressage is even worse. So the people, it's difficult to convince the people that you are investing in something to get something better in 10 years. The results are much more difficult to see, I think. That is one, I think, the most important.
Yeah, I think that you say about the stories of cloning these horses in Argentina, that you say, I didn't know, actually, I was listening with interest that is becoming popular. some way. And when you say these figures, about 10 years for the polo, even more for dressage, does it make very much sense to have you have to wait 10 years to have a good evaluation of an animal and then clone and then wait the other 10 years for that. Meanwhile, the genetic evolution, well, development of the population might probably make this animal that you clone an old animal in any aspect. Besides the problem, as you said before, mitochondrial, the environment, of course, will play a big role. So, but I found that cloning since the beginning, when it was coming out, and people, I remember 20 years ago, people were thinking, well, we can clone the best sires, and I remember there was a nice article by Alex, we are talking about by Van Vleck, say that is nonsense because of the biological time that you have to wait to evaluate the animals, then the other animals growing, and meanwhile you have the genetic development will make this genetic of the clone animal old. But I want to ask another question before time is running out to Tosso this time. Tosso, you listen with attention, as I'm sure I did. I'm sure you did, as I did for Sebastian's presentation, and also the last words about horses as a big companion animals. Do you see a lot of parallelism between the dog application of genomics in dogs and in horses?
Yeah, I mean, from, again, population structures, small breeds. So I'm not working in thoroughbreds, but I work also in some horse breeds, small local horse breeds, and the population structure and the science and the genetics is very similar between these horses and dogs. If I may say this in this audience, I perceive a more positive attitude among dog breeders towards genomics than what I perceive with horse breeders. So it's heterogeneous. One cannot really generalize, but with horse breeders, I perceive more resistance to adopt these technologies. Horse breeding is a very traditional business, and many horse breeders have very long-standing experience, which I don't want to be disrespectful at all. But yeah, I perceive a hesitance to adopt more science versus tradition-based experience in horse breeding than what I see in dog breeding.
Thank you, Tosso. Thank you. I think we are running out of time, and I want to be, at the time we decided to close the webinar at 5 o'clock. It's 1 minute to 5. So just the time to thanks Tosso Lab, Sebastián Demyda, Peyrás, and Alessandro Bagnato for these very interesting presentations. Thanks really much, very much. And it was a pleasure for me to listen to you, and I learned a lot today. And I thank everyone who participated to this meeting as as an auditor. And we had auditing. We had 180 person, and with the top participants 166. So a very large audience for a very interesting webinar. Thanks again. And EAAP webinar. This webinar, it was a question if we can see the presentation. The presentation will be shortly in the YouTube channel of EAAP available for everyone. So please, if you want to watch again or show it to some of your colleagues, they can find it there. And we have webinar every month, so stay tuned with the EAAP new things. 2 things we discussed today about companion animals a lot. I remember everyone, we have a workshop in companion animals in Milano between 14 and 16 of May. Go to the EAAP website and find the information. And artificial intelligence, there is a conference in Zurich between 4 and 6 of June. So thanks again to you, Alessandro, Tosso, and Sebastián, to everyone participating here. Bye-bye.
To you and to...
Thank you.
Thank you.
Bye.
Bye-bye.